Thanks for the nice introduction. I want to thank both organizers, Alex and Chris, for inviting me here. Oh, hi, nice to meet you. All right, so I come from the University of Utah, where I've been there since 2019. I started my research group based on biophysical modeling of biological macromolecules with an interest in adhesion proteins and more recently in microtubules. Here a little list of topics. I will start by speaking about how the sequence determines the structure of proteins. If we know how, I'll try to say something about that. And then once we know the structure, we tend to think that that is an indication of the function of the protein. For this statement, I will actually try to argue that the structural dynamics is much more important than the structure itself. And this is why my lab is running a lot of atomistic simulations based on molecular dynamics and enhanced methods for analyzing molecular motions. Those type of simulations run into several problems. One of them is the time scale. In order to be able to sample conformations above the hundreds of microseconds time scale, we actually do different things. One is coarse-grained in the system, so reducing the description, the number of degrees of freedom of those proteins. And in this case, we have been applying some machine learning methods to learn how to group the various degrees of freedom together and reduce their number. And then another method that we use to try to push the system along longer extended timescales is to build graph neural networks. So again, representing the protein in a more simple way with graphs, and use reinforcement learning to try to predict what happens at timescales that we can't simulate with full AtomMD simulations. I will present a few data from all AtomMD simulation of the adhesion protein integrin and microtubules. And then for the coarse-graining methods and the application of machine learning approaches, I will focus on microtubules simply because they are significantly larger than integrins. Instead of having 2,000 residues, they can span up to 250 micrometer length with a diameter of 25 nanometers, so millions of atoms. And so coarse graining and being able to predict their dynamics at extended timescales becomes more critical. And for the reinforcement learning and the graph neural network, I'll use the dimer, tubulin dimer as an example. All right, this is one of the slides that I present in my molecular biophysics class when I introduce protein folding. I show the ribosome and protein synthesis machinery, and then I end up with the idea that a random coil, a stochastic coil, actually rearranges into a three-dimensional well-defined structure, which is the native conformation. The fact that the sequence determines the structure is not novel, is not new to us. Amphisen actually proposes this dogma in around 1970s and got the Nobel Prize for this. Today we have AlphaFold 1, 2, 3, and also AlphaFold multimer that allows us to know from the sequence the structure of a protein. Those methods have ranged from deep learning techniques to diffusion-based generative models nowadays, and we are able to predict not only conformations, but also complexes and complexes of multiple proteins and proteins plus ligands and RNAs as well. Over the course of the last 10 years, 7, 8 years, those methods have increased their ability, not only in capturing the structure of single protein complexes and other macromolecular systems, but also they have higher and higher accuracy. In any case, what I want to argue is that if we start from a sequence of amino acids and we are totally capable of determining the native state of a protein, three-dimensional thermodynamically stable state, we still don't know much about the function, because within this native state, actually, there is a lot of subtleties, a lot more interesting things going on. For example, there could be multiple energy basins where the protein happily exists in its three-dimensional shape. And actually, those basins can determine the function more than the structure itself in some ways. What I mean is that if a protein goes from one basin to another in order to activate, that means that the function is fully governed by those transitions. And sometimes the difference between inactive and active states are not fully captured by only the sequence of amino acids. So this is why I do what I do. My lab is working on understanding the structural dynamics, because we believe that this is really what matters in determining the function of proteins. In particular, also, once we model proteins in different energy basins, what becomes particularly challenging is to cross this barrier of energy. And so some of the approaches that we use enhance the sampling by boosting energy into the system and giving enough energy for the protein to transition across conformations. So the simple paradigm that the sequence of amino acids determine the structure is somehow a simple right way to think about proteins, but the way we want to think about them is more dynamic and accounts for those for different states that can encompass also the same structure and obviously the same sequence. Those are examples of equilibrium MD simulations on this transmembrane receptor called integrin. Integrin exists in different shapes, in different structure. In the left here we see it bent, inactive, and on the right it's extended and active. What happens in those transitions from inactive to active is that integrin increases the affinity for ligand binding so that the cells can actually grab the external ligands that are in the extracellular matrix or in other cells and create adhesions in this way. In order to transition from inactive to active, integrin needs a boost of energy. So some of our simulations are under force in order to favor this transition. This transition is important for not only creating adhesions, but also for cells to migrate. And this is a schematic representation of how the leading edge of a cell can move forward. Integrins are here. They're colored with two colors representing the two chains, the alpha and the beta subunit. In the transition from inactive to active, integrin increases its affinity for ligand binding, so it can bind ligands, and on the intracellular side, it combines accessory proteins that bind to the actomyosin cytoskeleton, and from there, it can basically sustain enough traction to allow the actin filaments to polymerize, push the membrane forward, and therefore the cell ultimately to migrate. This is a mesoscale model based on those mechanisms. It implements the Brownian ratchet mechanism to accept pushing forces on the plasma membrane. And here in red, there are those integrins that become activated and create the connections to allow the filaments to remain stably bound and exert sufficient force for this motion. I said before that to transition from inactive to active, we need, in the case of integrin, external biases, external boost. And one way we simulate this transition is by adding external forces, running what is called the NITMB simulation, where we basically look at the flexibility of these proteins, and we are able to capture differences which are fully determined by the structural dynamics of these proteins. Differences between different beta-3 integrins, for example, and we found that alpha-2b beta-3 integrin, which is expressed in platelets, is actually more soft and more easily to unbend with respect to alpha-v beta-3 integrin, which is expressed in nucleated cells. Once integrin has transitioned between those conformations, we can apply other enhanced sampling methods in order to understand the changes in affinity. So we put a ligand and we try to see how the ligand binds, what is the free energy for ligand binding. For those type of simulation, we use what is called funnel metadynamics, where there is this funnel at the ligand binding site. and we change the position of the ligand to calculate the corresponding energy to identify where the minimum is. So the way those type of simulation look like is like this, where we are moving along the different reaction coordinates the ligand. And for each and every position, we calculate the energy. So we identify the minimum, and we are able to identify different binding poses and the contacts that can form. And by doing this in different integrin conformations, we can correlate how, by changing conformation, the ligand binding site changes, and so the binding affinity changes, and determine ligand binding propensity. A second way that we use to allow those transitions to happen from one state to another, so between two basins, is actually using coarse graining. So since all atom simulations allow us to simulate up to hundreds of nanoseconds or a few microseconds, depending on the size of the system that we are interested in actually reducing the descriptions of those all atom systems into few grains, and using potential energy functions that mimic the all atom system force field and relative fluctuations, we can extend both the size and the time scale of our simulations. So for this, I'll present the case of microtubules. Microtubules here have a hollow cylinder type of structure of 25 nanometer diameter. And in this case, we simulate 8 tubulin layers stacked in this geometry. The beta tubulin monomer is actually exposed at the pointed end and is the exchangeable side, the one that hydrolyzes GTP into GDP, and the alpha tubulin monomer instead is the one at the other end. So we are very interested into how those big polymers, rigid polymers, actually change shape, because this underlies the mechanism of chromosome separation and other processes in cells. Dynamic instability, it is what makes actually microtubules so dynamic, and their shape changes from being straight to being actually bent with the tips curved. During the phases of growth and shrinkage, typically in the classical model of dynamic instability, the GTP-bound dimers are at the plus end, and the shape, the geometry is actually straight. Then the GTP hydrolyzes into GDP, and this correlates with bent morphologies. This process goes on, and the phases of growth and shrinkage actually alternate. So why does that happen is the big question that lots of people have tried to address, and we are also interested in. So there are two contradicting models that, contradicting and complementary, I would say, models that try to explain this mechanism. One is the allosteric model that simply posits that GDP-bound tubulin dimers are bent. And this configuration basically weaken the lateral interaction between the protofilaments, so that as soon as GTP is hydrolyzed into GDP, the tubulin dimer bends, and this creates a weakening of the lateral contacts and therefore flared geometries. The second model is the lattice model, which instead doesn't go through this conformational change. It says that as soon as the GTP hydrolyzes GDP, the lateral interactions are weakened, and then the protofilament can bend in this way. There is a growing line of evidences that started in the last 10 to 15 years that actually shows that the GTP-bound microtubules are also bent, so they're not always straight. And this comes from cryo-electrotomography data that have been collected in vitro, but also from different cell types. The title of this paper is "Microtubules Grow by the Addiction of Bent GTP-bound Tubulins." So the distinction between bent and straight is not so much dictated from the nucleotide state. So in order to also try to gain some insights into this mechanism, we ran extended all-atom MD simulation on microtubules, and then we did coarse graining in this case. On one hand, we did Langevin dynamics by implementing what is called the iterative Boltzmann inversion method that I will explain. And then the second method that we did in collaboration with the University of Chicago is by using k-means clustering. And I will go over those methodologies. The second one is a unsupervised machine learning type of approach. So this is how our all-atom equilibrium MD simulation look like over 4 microseconds of simulation. Basically, the structures are not relaxed, and they relaxed over time. They tend to open and the protofilaments bend outwards. So if we calculate the root mean square displacement of all of the amino acids relative to their initial position, we can see that they keep moving relative to their initial position in the initial structure. So the system has not reached equilibrium. But if we then characterize some of the more microscopic changes that occur, we can find some structural metrics that equilibrate in 4 microseconds. This is the example of those angles, alpha, beta, alpha, and beta, alpha, beta, that are calculated from the center of geometry of the monomers. And we can see that those angles, for example, both in the GTP and in GDP-bound microtubules, tend to stabilize, specifically between 2 and 4 microseconds of simulation. If we look at the longitudinal distances between the center of geometry of nearby monomers, we see the same thing. The distance initially increases and then stabilizes over 4 microseconds of simulation. And similarly for the lateral interaction, except for those at the seam of the microtubule, where beta alpha and alpha beta contacts are established. So since those structural quantities seem to converge within 4 microsecond simulation, we use iterative Boltzmann inversion. So in other words, we start monitoring the distribution of those angles and distances. We convert those distributions into potential energy functions, which means we convert them into either harmonic potentials to establish harmonic oscillations between the beads that we are considering, meaning the monomers in this case. And we also convert them into Morse potential for all of the lateral interactions and the diagonal lateral interactions. The reason for this is that our microtubules will laterally separate, so we need a potential energy function that has a dissociation energy for large distance. So if 2 beads go too far apart, actually their energy doesn't go to infinite in those cases. And we use the Morse potentials for the lateral and the diagonal type of interactions. And then we apply this iterative process where initially we calculate the potential energy between all of the pairs of beads. And then we run a short coarse-grained simulation using Langevin dynamics to recalculate this potential. And we repeat this procedure as many times as needed until the radial distribution function of the distances or the angle or the dihedrals actually match the target being the one in molecular dynamics simulations that has stabilized between 2 and 4 microseconds. Then we run overdamped Langevin dynamics, where we basically update the position of our particles in the system based on the current position and the force acting on each and every particle divided by the friction coefficient, where the friction coefficient comes from the hydrodynamic radius of those beads, according to Stokes' law. This is actually overdamped Langevin dynamics and has this term here which represents the random noise. So in this type of simulations, we don't need explicit solvent. We have this stochastic contribution that add the effect of the solvent in terms of making particles fluctuating based on temperature. So we were able to extend our 4 microseconds simulation up to 44, so 1-fold longer, and characterize various structural properties, including the degree of bending between GTP and GDP-bound microtubules. up to 44 microseconds. First of all, we saw that the root mean square displacement of the beads relative to their initial position at the beginning of the simulation goes up and then saturates. So in this case, the structure seemed to reach an equilibrium state. And also we noticed that the GDP systems seem to deviate more from their initial configuration with respect to the GTP system, which is consistent with the idea that the GTP bound tubulins are those that are less stable, they polymerize faster, and potentially were believed to have even higher curvatures. If we measure this radial distance of the tip of the microtubule protofilaments from the center of the microtubule, we also notice that the system stabilizes and that the GDP bound systems seem to bend more. What we miss in this type of simplification of the system is actually a lot of details on the lateral interactions. The lateral interactions are actually consistent across all of the lattice. So we use k-means clustering, which is this unsupervised machine learning method that allow us to retain a lot of the potential energies between lateral tubulin dimers. After we redefine the grains with this approach, now we create an heteroelastic network. So we put springs that connect all of those beads based on matching the fluctuations from the atomistic system and run again the over-damped Langevin dynamics. I will say a few words about this k-means clustering algorithm. This is basically an extension of the essential dynamics coarse-grained method, which looks at where each and every atom of the system goes and cluster together things that move together. The difference between EDCG and k-means clustering is that the EDCG approach was basically clustering together residues and atoms of those residues that are consecutive in the sequence of amino acid. Whereas the k-means clustering basically lifts this restriction. It's able to cluster together things that are apart in the sequence of amino acids. So those are lines that design the sequence. And you can see here that the grains 1, 2, 3, and 4 can actually take residues from parts that are not consecutive, which is an improvement with respect to essential dynamics coarse graining. The algorithm itself is minimizing this loss function. It's trying to minimize in the fluctuations, so it puts together residues that move together. It tries to minimize the distance between them, so they also have to be close in space. And in addition to that, they try to maintain some continuity. But this is just to say it tries to minimize the probability that residues that are too far end up being together. However, they can still end up being together because of the distance criterion. And this is how the alpha and beta tubulin monomer look like with 20 beads each. And you can see here that some of those loop regions in the lateral surfaces of the monomers are actually discretized, remain there. So those details is what will allow us to capture differences in lateral interaction a little more carefully than with the other approach. In fact, our initial data have provided insights into the fact that GDP microtubules actually form more clusters with less protofilaments each with respect to GTP-bound microtubules. Also, the rate of bending is faster for the GDP-bound microtubules, which means that the bending angle over time is generally higher than the GTP. So this is consistent with the idea that GDP is less stable than GTP-bound lattices. Okay, and last, with this problem of time scales that we try to address with coarse graining and different types of Langevin dynamics built on different systems, we are basically facing the question of training a network and based on a sequence of states of the system at each time point, trying to predict what happens at the next time point. And if we are able to do that, it means that we can, in principle, keep applying this method and predict infinite time scale or milliseconds time scale. Let's give it some finite idea. So to do so, we are taking the tubulin dimers, we are building this graph neural network, defining nodes and edges. We're using protein language model in order to embed those nodes. And then our current architecture has basically those two components: proximal policy optimization component, which is used to trade the framework to make some decisions, and then this discriminator, which pertain to the adversarial inverse reinforcement learning. In other words, this type of approach is looking at the data from all atom simulation and trying to learn the rules, and gives rewards based on the learned rules. So this is all work in progress, and this is pretty much where we are with our strategy in terms of like building a new net. This is something that a few other labs have tried to do, and we'll see how our network compares with them in hopefully in the next few months. So I want to summarize what I spoke about today. I started with this relationship between sequence and structure, showed that AlphaFold now allows us to start from a sequence of amino acid and predict a structure, but the structure determines the function of a protein, but is still, there could be more things involved, meaning the structural dynamics. I show you the examples of integrin undergoing structural dynamics at equilibrium and under force through transition between conformations, and also the microtubules at the all atom level. They tend to bend and tend to try to reach a more stable configuration. Both those systems face the limitations of the time scale. The time scale accessible by molecular dynamics simulation is still below hundreds of microseconds time scale, and real biological processes can occur over seconds to minutes. So there are different methods to go about this, and some of them involve machine learning and deep neural networks. For coarse graining, I presented those two approaches: the iterative Boltzmann inversion method-based approach and the k-means clustering approach, and a representation of the architecture that we are trying to develop to achieve longer time scales for the study of tubulin dimers. With this, I would like to thank you for your attention. I'll be happy to take any questions.
Plenty of time for your question. I have two questions. For the all atom simulation, seems it's achieved very long time scale. Did you use explicit solvent or implicit solvent? Or
explicit. We use what is called TIP3.
Using big machines to
Yeah. So for the integrin system, we have a number of atoms of the order of 500,000 to 800,000. For the microtubules, the number of atoms goes up to 30 million atoms. So we had, for this type of simulation, the approach was a little different because we created a box, a water molecule plus the protein. The protein starts bending so that the tip of the microtubule pass over the surface of the box. So we had to regenerate a new box, add new water. So we started with 21 million atoms up to 30 million atoms, and the type of water molecule is a 3-point water molecule with 3 beads that determine hydrogen, and oxygen of the water.
So the second question seems is you used each CG site for each tubulin monomer, right?
Yeah.
So there's a bond angle dihedral interaction between them.
We use a total of 13, let's call them structural details, and we have dihedrals that encompass 4 beads on 2 neighboring protofilaments as well.
So my question is, some people believe the longitudinal strength stiffness is very larger than circumferential. Maybe you say even 1,000 times larger, because you definitely using the inverse, iterative inverse, Boltzmann inverse, you get some coefficient. Is that the case? Because, for example, you can compare the bond strength in longitudinal versus circumferential. Do they show such major difference? Because usually they say it's very anisotropic in longitudinal versus Well,
yeah, I have some insights on that. Yeah, I think in those simulations, they are the longitudinal interactions are stronger than the lateral interactions. In fact, we see that the breakage basically of those bonds laterally. I know that those things depends on the compaction level. So microtubules are more compact in the interior parts. This is why GTP is hydrolyzed faster there than outside. And there is a balance that has been proposed between lateral and longitudinal interactions. One of the last simulations that I've seen actually have shown that the longitudinal become also pretty weak in different ways depending on the GTP versus GDP bound state. But in those simulations that I presented here, the longitudinal interactions are stronger than the lateral interactions, and they are pretty consistent between GTP and GDP. There are different answers to these questions. Hopefully I addressed it. Do you have any further insight you want to discuss?
Yeah, I agree.
Okay.
Other questions? I'll ask one. Can you explain a bit more the intuition of the reinforcement learning approach? As in, you have like a gold standard all atom simulation and you're trying to learn some effective rules. Can you explain why you need like all three things and like what role they play?
Because we want to compute the difference between those two. So we are trying with this proximal policy optimization from basically trial and error to understand what makes the trajectory not to have an error, but going into the right configuration. And then with the discriminator, we learn what is the reward function. So if we have one right thing and also one right reward, and we put those two things together, hopefully we get a better answer with higher accuracy. Yeah, this is actually something we are now testing on these tubulin dimers. And yeah, there will be more to follow.
All right, let's thank her again.